Hi everyone, we're so glad that you've joined us at the Center for Brain Health for our event. Let's get started with a brief video underscoring the importance of brain health for you and for society. After that, we'll introduce our speaker.
We work, we live, we innovate and create. At the center of it all is your brain health. The ability to solve problems, think analytically, share empathy, and thrive.
We're trying to make brain performance really the next fitness revolution.
So how do you boost brain power? Welcome to the Brain Health Project, an urgent call to transform your mind to work stronger and faster.
This is an absolute crisis, as great as any we have ever faced. We have to equip the minds and brains of our citizens to cope with the accelerating, dizzying rate of change that they face in their lives.
Your brain health is not fixed. Scientific discoveries prove it can adapt and grow regardless of your starting point.
Our greatest value, the asset that will help us to change everything, every problem we're in, is all in our head.
To harness that treasure, we must measure and monitor progress while things are going well versus waiting for an injury or disease to strike.
Too many of us are outliving our brains, and that does not have to be the case.
The information age is bombarding us with more content than our human brains can handle. How do you keep from getting lost in this? And focus on deep thinking. For starters, stop multitasking.
Science shows us that multitasking is bad for your brain. It reduces fluid intelligence, causes brain atrophy, and increases chronic stress.
The global pandemic is creating more stress than ever, stress that leads to depression and anxiety and beyond. Unlocking our potential to navigate these hurdles starts with learning the right strategies, even in school.
So when teachers have these strategies, they're empowered to support our learners, and then the learners are now able to take ownership of their learning.
Training kids how to think is doubling academic achievement among middle schoolers.
I think the greatest national security threat is pre-K through 12. If we don't take care of educating our young men and women, then we have to ask ourselves, where are we going to be in 20 years?
Our world-renowned scientists know you can increase your brain health, not lose it. It's time for a new category of health, brain health. You are a game changer. Ready to transform the world with us? Be a part of the brain health revolution. It starts here.
Okay, welcome everybody. We'll go ahead and get started in just a moment. This is, of course, the Frontiers in Brain Health series, where we take a deep dive into some of the most exciting science related to neuroplasticity, cognition, and behavior. I'm Dan Krosik, the Deputy Director here. We are a cognitive neuroscience center dedicated toward exploring, protecting, and healing the brain. And today we have a very exciting scholar in the areas of attention and memory, Dr. Matthew Robison. And Matt did his bachelor's at Notre Dame. He was actually a dual major in psychology and business and entrepreneurship, which I had learned just today. He then went on to do a PhD at the University of Oregon, working with Nash Unsworth. And this has to be among the most prolific mentoring relationships I've ever seen. I counted that they've published 32 papers together to date. That's just unbelievable. So remarkable collaborations. And then Matt went on to do a postdoc at Arizona State, working with Gene Brewer. Now, since 2020, he's been at nearby UT Arlington, working on his assistant professor career there. And I was really amazed when Matt Robison, when I learned he was in the DFW area, because I'd actually known of him from a paper, a really influential paper by Nash Unsworth and Matt Robison in 2017 in Psychonomic Bulletin and Review. This is one of those papers that you just think of in your career. It's one of the most impactful papers I've ever read. And the reason I say that is just it bound together really key concepts for me, like how attention and working memory function, and how we can think about memory capacity in relation to attention. It also talks about norepinephrine and Pupilometry and how we can use that as a measurement of arousal and how that impacts our attention and working memory. And then furthermore, it linked together the task mode brain activation and the default mode brain activation. So just a tour de force of integration across all these interesting, fascinating measures that are coming around, with the centerpiece being really Pupilometry measures. And that's just an amazing way to measure attention and arousal. And Matt's been doing some of the most careful work in this area, which he's going to talk about today, learning about how we can basically measure cognitive abilities in relation to that all-important biomarker, which gives us a real window into the mind. So I was so excited to have Matt Robison in the DFW area, and we connected, and what a difficult time to start your professorship right during the COVID year. But he's now collecting in-person human data, and things are rolling, and he's got Pupilometry measures going. So we look very much forward to hearing this talk. For those of you joining us virtually, please use the Q&A function if you have questions. We'll get to some of those at the end. And for the in-person audience, you're welcome to ask your questions also at that time. So without further ado, welcome Matt Robison.
Thank you. That was a very, very nice introduction. It's great to be here. The center is phenomenal. This is my first time on campus, and I'm pretty close by. I live in Dallas. I'm just on the other side of Love Airport, so really cool to see the facilities. I'm looking forward to seeing more and meeting more of you during my visit. But yeah, today I'm going to be talking about some of the work I've done towards the middle and end of my graduate career, and some of the ongoing and future work that we're doing in my lab at UTA. And so I'll just start by giving you an overview of my research questions, and my clicker's not working, that's okay. Then we're going to get into some specific topics focusing on attention and working memory. Then I'm going to share a bit about some of the work that we are currently doing and some of the work that we're really excited about doing in the next year or 2 years. So one of the more fundamental questions that I'm just fascinated by is why it feels difficult and effortful to sustain and control our attention, and why, as human beings, this is something that appears to be such a limit for us. And especially, it feels particularly difficult. Maybe it's not more difficult, just feels more difficult based on the abundance of information that competes for our attention in our daily lives. But if you've ever sat through a long research talk or meeting, or just done the same task for a long period of time, you've noticed that it just feels hard to do something for a long period of time and pay attention. And so that's a very basic thing that I'm interested in. But today, I'm going to be focusing on these last two questions, which are, why do people differ in their attention and memory abilities? And what might be the brain systems that underlie those individual differences? Just like most human traits, cognitive ability is sort of normally distributed in the population, and we just want to understand where that variability comes from, and leveraging different tools to understand what gives rise to those differences. So first I'm going to talk about the specific case of controlling a person controlling their attention. So this is a little diagram of me going about my day. Sitting at my computer, and I've got a to-do list, but all the time, my phone's buzzing, People are calling me, research assistants are popping by my office with issues. My wife is calling me to talk about carpet, and there's all of these things competing for my attention, and these are sources of what I call external distraction, right? Things that are happening outside of me in my environment that cause me to lose attentional focus. But we also know that lapses of attention can happen from internal sources of distraction, or what I call mind wandering. So something that's been on my mind recently is I wonder who's going to win the NBA Finals. The playoffs are going on right now, and that somehow occupies way too much space in my head, but it is something that competes for my attention when I'm trying to get my to-do list done. So I'm going to build off of a framework today that we've been working on over the last couple of years that implies that there's at least two important sources of variance across people that drive individual differences in attention control and, in this case, working memory. And these are intensity and consistency. So there's this famous quote from Bruce Lee. I don't even know if he ever said it, but it's attributed to him that long-term consistency beats short-term intensity. And hopefully what I can convince you by the end of the talk is that he's wrong. Both intensity and consistency are important, and in different ways, at least when it comes to mental effort. Maybe he was right about martial arts. So both of these play into how well our attention operates, how our memory systems operate. The problem with intensity and consistency, and I guess I should quickly define what I mean by those. By intensity, I mean the sort of aggregate or absolute amount of mental effort someone is applying to a task at any given moment, and sort of the average amount of effort they give. Whereas consistency is the moment-to-moment regularity with which they do that, right? So you can be intense in a given moment, but if that's not consistent, then that's not going to behoove you as much. The problem with intensity and consistency is that, like most psychological features of individuals, they're hard to observe, right? These are inherently internal processes, and so we have to design clever ways of actually measuring them and objectifying them. And so that brings in the tool that I've been using pretty extensively in my research, which is pupillometry. And as you can probably tell from the name, pupillometry is just measuring the diameter of the pupil on a moment-to-moment basis. Obviously, the main role of the pupil is to control the amount of light that hits your eyes. It constricts when we're in bright conditions, it dilates when we're in dark conditions, and allows us to be adaptive based on the environmental luminance that we're experiencing. But we've also known for a fairly long time now that it dilates for other reasons, right? It's connected in some way to the sympathetic nervous system, so when you have that arousal response or that fight or flight response, a typical corollary of that is a pupillary dilation. So it's not just about how much light is allowed to hit the back of the eye. I'm going to talk about some new work that I've been doing, but pupillometry and cognitive pupillometry specifically, it's actually a very old technique, and it goes all the way back to the 1960s. So this was probably the foundational study that allowed people to start thinking about how to use the diameter of the pupil as a way of studying human cognition. And this is what they did. They just had, I think there was 5 participants. This was the 1960s, so 5 participants. They did 4 trials, and they just had people complete mental math problems while they took pictures of these people's eyes while they did those math problems, and then eventually gave a response. And so they had each of the 5 participants complete 4 different math problems that scaled in complexity or difficulty. And what they found was that the magnitude of the pupillary dilation compared to a baseline measure that happened a few seconds before they saw the math problem, it scaled with the difficulty of that problem. And so what they determined, or they surmised, was that the pupil can kind of be used as this global measure of mental effort. What that was actually indexing at the level of the brain, they were kind of agnostic about, but this really launched the field of cognitive pupillometry. And interestingly, cognitive pupillometry kind of died out for 70s, 80s. If you go back and look for papers, there's a few from people like Jackson Beatty, Dan Kahneman had a whole book about had a book, his Attention and Effort book, pretty extensively yet leveraged this technique to look at theories of cognitive capacity and mental effort, but it's really picked up recently, and we are kind of riding that wave. So this is what we do in my lab. We've got more sophisticated eye trackers. These eye trackers that we use collect data from each eye at about 150 times per second, and we just continuously measure the diameter of the pupil as people are doing our tasks, the computerized tasks of attention and memory. So we get a readout of the pupil, and we can time lock the responsiveness of the pupil to various events that happen or cognitive activities that people are performing. So here's a task that we use a lot in my lab. It's called the Psychomotor Vigilance task, and it actually is borrowed from the sleep deprivation literature and the sleep literature in general. And all it asks people to do is look at this row of zeros in the middle of the screen, and after an indeterminate and unpredictable amount of time, those zeros are going to turn on like a stopwatch, and you just have to hit a key, usually the space bar, as quickly as you can when you notice that happening. So this is what it looks like. When they press the space bar, the numbers turn red. It gives them a little bit of feedback about what their reaction time was, and then they go on to the next trial. What makes this task difficult is two things. One, it's really boring. So the longer you do it, the harder and harder it gets to sustain and control your attention to the task. It's very easy to slip into mind wandering, to get distracted by things happening around you. The other thing I like about it is it's very simple, right? People can understand the instructions on one practice trial, right? It doesn't require any skill development, and in fact, there's really no practice effects. The longer you do this task, you're not really going to get better at it. So you can give it to someone, you can give it to virtually anyone, and they get it, they understand it, and on every trial, which happens anywhere from 3 to 10 or 12 seconds, we get a readout of their attentiveness, which is a very simple measure, their reaction time. A long reaction time probably means they weren't paying very much attention. A fast reaction time means intact attention. So what we do then is we take everyone's reaction times. In the experiment that I'm going to present, people did 140 trials, which took about 30 minutes. And it's brutal. It's so hard to do this task for 30 minutes. And I ran 350 of these people. They were all undergraduates for my dissertation, and they hated me by the end of it. But what we do is we take their reaction times on every individual trial, and we rank order them from fastest to slowest. And what I'm trying to show you in this graph is that the variability across people shows up at that slow end of the distribution. So when people are at their fastest, they're really about equal. It's when they're at their slowest, when they're experiencing what I'm going to call attentional lapses, when those individual differences really pop out. And it's the individual difference that we're interested in is the frequency and severity of those lapses. So a more severe lapse will be an even longer slow reaction time, and frequent lapses obviously are going to be lots of long reaction times. So what we do is we take, as our global measure, the individual difference, we take the 25 slowest trials, and we average those together. And that's our dependent measure from this task. There's lots of other dependent measures you can get from it. You can use mean RT, you can use median RT, you can use variability in reaction times, but we just use this slowest quintile of trials from the distribution. So as they're going through this task, we're collecting pupil diameter. And what we get are two measures that we're specifically interested in. In the eye tracking version of this task, we throw a fixation screen in between every trial. And the fixation screen is exactly how it looks. It's just a row of fixation crosses, and all they have to do is stare straight ahead at those. That gives us a sort of moment-to-moment measure, trial-to-trial measure of their arousal state. And as you might imagine, arousal state fluctuates just like anything else from moment to moment, just like attentional state. And so the measure that we are extracting here is just the mean over that 2-second window. For each trial, we use to compute a consistency score. And so more consistent individuals will show very little variability, and more inconsistent individuals will show lots of variability. And I'll show you a couple plots that show that. And then the second thing that we're interested in is the task-evoked pupillary response. So they're waiting, and then boom, the numbers start counting. What happens is the pupil shows this really quick blip, and we use that as our measure of how intensely people are focusing in that moment. So what I'm plotting here are two individuals. This is real data from two people who I've pre-selected as a consistent person and an inconsistent person. The consistent person, you'll notice, they've got some fluctuations from trial to trial, but it's really not on a large scale. Whereas this person on the right, their pupil diameter is really bouncing around. And if you were to extract the mean of those values, they're actually about the same. But where the differences between these two people pop up is in that consistency measure, the variability within an individual. So what we do for each person is we calculate the coefficient of variation, which is just the standard deviation adjusted for the mean, and that is a value that we use as an individual difference. How consistent is a person's pupil diameter before a trial from trial to trial? So higher values for that coefficient of variation mean less consistency or more inconsistency. Then to quantify intensity, this is that pupillary response that I was talking about. It shows this really quick blip that comes right back down at the end of a trial, and we just compute the average magnitude of that as our measure of intensity. So for each individual, we've got two scores derived from the pupil data, a consistency score and an intensity score. And what we're interested in is whether these things predict ultimately how well people perform on the task. So when we look at a correlation first between our inconsistency score and our slowest 20% of trials, we get a positive correlation. So people who are more inconsistent in their arousal state, moment to moment, do worse on the task. So that seems to be important. When we look at intensity, we find the opposite pattern. People who are more intensely focusing, as measured by the average task-evoked response, do better on the task. They have faster reaction times. And so greater intensity tends to correlate with better attention control. So inconsistency, worse attention control; intensity, better attention control. So both intensity and consistency seem to be important. An interesting question then to ask is, well, to what degree do intensity and consistency correlate? Is it just the case that intense people are consistent and inconsistent people are not intense? Very weak correlation between those two things. In this case, we've got about 350 people in this sample, so that correlation of 0.14 is statistically significant with an alpha of less than 0.05, but or a p-value of less than 0.05, but it's small, right? So what we conclude here is that consistency and intensity, while both important in predicting performance, appear to be rather distinct individual differences. And I think that might be an important thing to think about moving forward. So just to quickly summarize, what I'm hoping to convince you is that we can use pupil diameter to measure both the intensity and consistency of attention, that people who attend more intensely and consistently avoid frequent and severe attentional lapses. I'm going to quickly, I'm going to move now to working memory, which is a related cognitive construct, but slightly different than attention control. So many of you here might be familiar with the concept of working memory, but in general, what working memory is our ability to handle, manipulate, actively maintain multiple pieces of information in a short period of time, right? This is sort of the activated portion of conscious awareness that we have. But what we know is that this is limited, right? We don't have an infinite ability to handle information at any given time. And it's probably an adaptive feature, but it is a limitation of the human cognitive system. What we also know, though, is just like attention control, working memory capacity really varies across individuals. Some people have really high working memory capacity and some don't. And what got people really interested in working memory capacity as an individual difference is this finding that started popping up in the 80s and 90s that it was predictive of all kinds of things. People with higher working memory capacity were doing better on measures of intelligence. They had higher SAT and ACT scores, GPAs, GRE scores. They were learning languages faster. They had better reading comprehension. And so this led people to get really interested in working memory capacity as a construct. So there's actually a long history of research on individual differences in working memory capacity. So what we decided to do was throw our eye tracker in front of people as they did a working memory task to see if that same pattern of correlations popped up. So what we did was we gave people this very simple task, which was, I think, first published by Luck and Vogel, Steve Luck and Ed Vogel. And what it does is it presents people with a pattern of colored squares, or it could be other items, oriented bars, shapes, things like that. It presents them very quickly, it takes them away for a second, and it brings them back. Usually one of the items is circled, and people are asked, is that item the same or different as it was when you first saw it the first time? So I'm going to need a little audience participation here. I'm going to give you this task, and I want you to verbally tell me whether you think that on the test screen the circled item is the same or different. Okay, here we go. Nice work. All right, another trial. Different. Another trial. Pretty good. All right, a couple more trials. Pretty good group. All right, I think everyone did really well there. But what I was hoping to show with that little demonstration is that as the amount of information increases, the task gets considerably harder. And in fact, at a certain point, it becomes almost impossible to remember all of the items. And what Luck and Vogel argued with this original paper was that this is demonstrative of the fundamental capacity limit of working memory. There's only so much information we can hold in mind at any given time, especially over short intervals. You know, long-term memory is unlimited. There's no real limitation that we've been able to reach on our ability to learn information and produce that information, but over very short intervals, it's definitely limited. So what we do is, just like the psychomotor vigilance task, we get those same two measures, pre-trial pupil diameter and the magnitude of the task-evoked response. In this case, the task-evoked response is coming from the delay. So what we do is we stretch the delay out from a second or two to 4 seconds to allow the pupil, which is actually a pretty sluggish signal, to do its thing. Doesn't really change performance on the task to make that delay longer. It might change strategies a bit, but it allows us to extract this intensity signal. So this is what that signal looks like. As people are trying to remember those items over that 4-second delay, their pupil dilates, and it sustains that dilation until we test them. So what we think that's indexing is this effortful allocation of attention toward that memory over that interval. So that's our measure in this case of the task-evoked response, which is our measure of intensity. The consistency measure is exactly the same as we did on the psychomotor vigilance task, trial-to-trial variability in the pre-trial measure. Here's the correlation between the variability or consistency measure and working memory capacity. Again, it's negative in the sense that more inconsistent individuals have lower working memory capacity, just like they had poorer attention control. And people who more intensely attend, as indexed by that task-evoked pupillary response during the delay, tend to have higher working memory capacity. So again, we're seeing the same exact pattern of correlations between our consistency and intensity measure when we move from a very simple attention task to a little bit more difficult but manageable working memory task. And again, we see virtually no correlation between intensity and consistency. Again, driving home the point that perhaps these are two fundamentally different aspects of people that are both important for determining their performance. So in general, what we're learning is that consistency and intensity are both important correlates of individual differences in attention control and working memory capacity. If anything, intensity and consistency are weakly correlated, but definitely two different things. And I just want to note that these are not the only two instances where we found this pattern of correlations. Pretty much any time we have someone do a cognitively demanding task, whether it be another measure of attention, another measure of working memory, or a long-term memory task, which is my most recent publication on this, people show the same pattern of relationships. Inconsistency in pretrial pupil diameter is indicative of worse performance, and the intensity of attention over some encoding window or memory window or response window seems to be positively correlated to their performance. So we think this is a sort of domain general feature of people that's important for a lot of different cognitive abilities. We've focused mostly on attention and memory, but we think maybe there might be more applications that we could explore in the future. So since this is the Center for Brain Health, you're probably wondering, what does this have to do with the brain? Is there any way that we can tie what we're observing from the pupil to a brain system? And as Dan was mentioning in the introduction, we think this might be indicative of differences in the Locus Aurelius norepinephrine system. So the Locus Aurelius is a brainstem set of nuclei that sit in sort of the low back of your head. And they're very small. I think it's 20,000 neurons or 40,000 neurons. But they release a lot of the norepinephrine or noradrenaline into the brain, which is this energizing neuromodulatory thing that allows us to do these kinds of computations. And what we think might be happening is that in some way, this system gets dysregulated. This is just a theory. We don't have good neurological or neuroimaging evidence for this, but we're hoping to get some soon. But our hypothesis is that somewhere along the line, the connection between the Locus Aurelius noradrenergic system and these higher level cortical areas like the frontoparietal control network, the default mode network, the salience network, which are the areas that we think implement things like attention control and working memory, somewhere along the line that's getting disrupted. What exactly is causing that disruption, we don't know, but we're interested in finding out. So one of the immediate future directions for this line of research, I think, is trying to figure out what this really might mean at the level of the brain. So the first thing that we're hoping to do, and right now we're trying to secure funding to actually do this project on a large scale, but is actually image the Locus Aurelius at an individual person level. So because of the Locus Aurelius' size, it's very small, where it sits in the brain, it sits right next to some major arteries. So those of you who know much more about neuroimaging than I do know that that's an issue with movement. As the blood pumps into the brain, the Locus Aurelius actually bounces around, which is a problem for neuroimaging. What people have learned recently about neurons in the Locus Aurelius is that they contain something called neuromelanin. And you can build a neuromelanin contrast into a neuroimaging scan to isolate neurons that have neuromelanin from neurons that don't, which allows us to show these very small, and I'm trying to highlight them as best as I can, dots in the brainstem. And this is a long slice going this way, and this up here, you can't see my cursor. But anyway, what I'm trying to show is that we can image the LC, and we can extract several important features of LC functioning from just this resting state scan. So all people are doing is sitting in the scanner and left to their own devices. This bottom panel here, the red line, or the red bar, what that's showing is that Locus Aurelius neurons just kind of fluctuate in their firing from moment to moment, very similarly to how pupil diameter just tends to fluctuate trial to trial, moment to moment. And we think that maybe that variability might be one of the things that's causing the inconsistency measure that we're observing. The other thing that we've been able to extract is a bold response from neurons in the locus Aurelius when people are doing an attention task. And that's what this figure up here, the figure B, is showing, that when people are given a stimulus to respond to, we get this slow but robust, bold response out of the LC. So potentially, we could remap our consistency and intensity measures off of pupil diameter and directly onto the LC using this neuromelanin sensitive scan. So it's a long-term goal of ours to do this. We've not gotten the funding to do it yet, but we're actively seeking it. The other thing that I'm interested in, and this is much easier to do because I can do it with undergraduates in my lab, is try to understand the role of things like motivation, right? Maybe this is just motivation. Maybe it's just the case that some people come into the lab, they want to do well, they're intrinsically motivated, they want to impress the researcher, and so they try. And the other people are just not wanting to be there, they're not motivated, and so when I'm measuring intensity and consistency, those are just manifestations of individual differences in intrinsic motivation. So what I've been trying to do is push motivation around to see how that affects intensity and consistency. And what I found, and this is a very simple design where all I did was give people points in a version of the psychomotor vigilance task based on how quickly they responded. So on every trial, if they were particularly fast, they got 50 points. If they had a sort of middling reaction time, they got 10 points. And if they had a slow reaction time, they got 0. And at every couple trials, I would give them their point total, and the points didn't mean anything. It was like, whose line is it anyway? They didn't get a reward. They couldn't cash those in for a prize. It was just try to get your point total high. And so we had a points condition and a no points condition. And what I'm plotting here on this black line is the no points condition. And this is a very stereotypical effect in this task that performance just gets worse and worse and worse the longer people do it. This is a half hour worth of doing this task. Pretty typical. When I just give people points, look what happens. Faster overall, and I've gotten rid of that time on task effect. So the question became, we ran this study online for obvious reasons, and then recently we did it in person when we were allowed to get the pupillary signatures. And I'm embarrassed to say I have yet to look at the consistency data because there's slight differences in the, we just finished getting this data a couple days ago. But look at the difference in the task evoked response here. So this is the points condition, this dotted line, and this is the no points condition. This is a difference of almost 3 times in the magnitude. So clearly, we can push intensity around experimentally. What that means for individual differences, we're not quite sure yet. One thing I can tell you is that when you try to measure intrinsic motivation, you just ask people, like, how motivated did you feel to do these tasks? They correlate with intensity and consistency, but very weakly, in the neighborhood of 0.18 to 0.25 in my studies. And they do not mediate the relationship between the intensity pupillary measures and performance. So it's clearly not just motivation, but the fact that we can push motivation around experimentally might allow us to do some interesting things down the road. Now, the second thing that I'm interested in pursuing is investigating the roles of things like sleep, physical health, and aging. So we just started doing a study a couple weeks ago where people come into the lab, we have them do attention and memory tasks while we measure their pupil diameter, and at the end of the session, we give them a Fitbit. And they wear it for 2 weeks, and they come back to the lab, and they do that same battery of tasks again. And not quite the same, but another battery of tasks. And what we're interested in is whether someone's sleep habits, their physical health, how active are they on a day-to-day basis, whether that has anything to do with the measures that we're observing, both cognitive performance and these intensity and consistency measures that we get from the eye tracker. So just here's the data that we can get. We can get lots of different sleep statistics. We can get the regularity of their bed and wake times, which seems to be important. We can get the sleep efficiency, so how much time are they spending in deep sleep and REM sleep versus light sleep or awake. And we can get kind of average measures of those things. So night-to-night variability and then kind of average over that 2-week period. Now, 2 weeks is not a very long period of time, but it's a long period of time to get an undergraduate to stay engaged with a study, and I'm just worried that if we stretched it out any longer, they'd forget they were even in the study and not return the Fitbit, which has happened once already. The other thing that we've been doing recently is looking at how these effects change with cognitive aging. So what I'm plotting here is the task-evoked pupillary response from the psychomotor vigilance task for older and younger adults. This blue line here is younger adults. This orange line is the older adults. And there's a couple different things I want you to notice from this plot. One, older adults' intensity of attention is actually greater than younger adults. They're trying harder, and in fact, there's a big confound in our studies between motivation and age, because the older adults that come into our lab way more motivated to do well on these tasks than the younger adults. But here's an interesting thing too, is look at where these dilations peak. There's about a 150 to 200 millisecond difference between the peak or the latency of that response and older and younger adults. And one of the most profound effects in cognitive aging is the speed of processing difference. And so this could be an easy to collect measure of a processing speed difference between older and younger adults. Now, there are other explanations for this. It could be that the pupillary dilator muscle is just differentially effective for older and younger adults, and we're doing some follow-up work to figure out whether that's the case. But when we saw that, we were really impressed. And we published this just a couple weeks ago in Psychology and Aging, if you're interested in reading more. The other thing that I want you to see is the behavioral data. So I've separately plotted older and younger adults. Again, younger adults are the blue line, and typical pattern of performance. They're getting worse and worse and worse as they do the task. The older adults, although they're slower overall, actually sustaining their attention much better. This line is statistically flat. And this was the first time before I ran those motivational manipulations that I ever saw a flat line at the group level in this psychomotor vigilance task. It always looks like this. And so when I saw that, I was like, wow, older adults, this might be something that they can actually do. They are actually better than their younger adult counterparts at, which is sustaining their attention despite being slightly slower overall. These adults were between the ages of like 65 and 83. I think the average was about 71. Pretty sure that's the average, but yeah, they were all older than 65. Okay, so the third thing that we are interested in, and this is obviously a lot of people in this room are probably interested in this too, is can we modify cognitive performance via intervention? What I'm specifically interested in is pushing around these intensity and consistency indices, which I think might allow us to be a little bit more, we can start to get more mechanistic about which of these effects we can or can't improve. Maybe we can improve consistency but not intensity, or vice versa. So 3 things that we are looking at are physical fitness interventions. We don't have any of these currently going. This is a future direction, but these are areas that we are writing grant proposals around and looking for collaborators and things like that. Sleep quality. So I mentioned sleep earlier as a potential correlate of these things, but if we can, experimentally improve someone's sleep quality via various interventions, seeing the effect that that has on these cognitive outcomes. And then another thing that I've been trying to do is leverage the pupil as a biofeedback mechanism, right? So if we can kind of track someone's moment-to-moment alertness, arousal, cognitive performance the intensity of their attention from moment to moment, can we design a system that would covertly monitor a person's attentiveness and do something to intervene on a moment-to-moment basis when the algorithm, so to speak, catches them in a moment of attentional lapse? Can we design a system that would bring attentiveness back online with, again, a covert measure of their attentiveness, perhaps even without them having to register a reaction time or an accuracy on a trial? I recently got some funding from the Navy to do this kind of thing in combination with some of the individual differences work I'm doing. So we're really excited to pursue that here in the next 12 months or so. All right, so with that, I just want to thank everyone. I do want to acknowledge all the people who are part of my lab and my funding source. I have a huge team of undergraduate research assistants. They're fantastic. And then my new graduate research assistant, Stephen, he just got this Naval Research Enterprise internship. So he's going to work with the Navy Research Labs in DC this summer. So I just want to brag a second about his accomplishments as well. But thank you all, and I'm happy to take questions at this point.
Okay, so we have some time for questions, clearly. Go ahead, Sandy. Yeah, I guess while we're getting a mic over there, one thing that jumped to mind for me, really fascinating work on the intensity and the consistency. And I've sometimes thought about people as kind of like intellectual sprinters versus intellectual marathoners. And I just wonder about phenotypes. I was imagining maybe 4 phenotypes because you said those weren't really correlated. I wonder if there's any evidence or thoughts about that maybe some people are intense but don't have consistency, others are very consistent but don't have intensity like the marathoners, and then you would imagine the other two where some magical people are intense and consistent, and then some unfortunate people are very inconsistent and not very intense and maybe really chilled out. And I wonder if if there's any merit to that, and that might be why there's not a tight correlation.
Yeah, this is an interesting question. So I haven't really done any subgroup analyses. This is something that Nash Unsworth has done a lot in his work. But what I can tell you is that very recently so the data that I presented from the Psychomotor Vigilance task is a big data set that I collected on about 350 people as part of my dissertation. And they all did various iterations of that Psychomotor Vigilance task. And what I was manipulating was various things to try and push performance around. But what I ended up with was this huge consortium of data, behavioral data. I had mind-wandering reports. I had motivation ratings. I had reaction times, pupil diameter. So I've been analyzing the heck out of that data over the last couple of years. And one thing I found recently, which kind of took me by surprise, was that people who start out at a faster level of performance, a better level of performance, they actually show larger changes in larger worsening of performance across time, whereas the people who kind of start out at a middling level tend to stay pretty flat. And so this might be exactly what you're talking about, where those flat individuals are the marathoners. They can pace themselves, so to speak, across the course of the task. And the people who are really going for it early on and then they just start to lose steam and their reaction times get slower as they go through the task, maybe those are your sprinters. So we've been doing some like linear mixed effects modeling and longitudinal growth curve modeling on the data to try and parse apart sort of initial performance and changes in performance and what those might differentially correlate with.
Okay, fascinating. Yeah, Sandy.
Matt, really very fascinating work and relevant to what we're doing. One of our collaborators on the BrainHealth project, Ian Robison, is very interested. I don't know if you're familiar with some of his work.
Oh, yeah.
And Locus, Aurelius. So I'm like, we've got to hop on a call today with him because this will make his heart beat faster and excited. I have a number of questions. One is with your, and I don't know if it's with the Navy or what you proposed, are you thinking that these elite performers that are called to do things that maybe you could help fine-tune the system with the feedback in real time to you've lost attention, so snap it back real quick for more elite performers in the special ops forces, for example?
Yeah,
Was
so
one question, but I have two.
Okay, I'll let you ask. Do you want me to answer the first one first? Okay, so the long-term goal of these biofeedback programs, which I have to be honest, we don't have yet, but we're working on designing, would be to train people to do that on their own, right? So there's this metacognitive ability to be able to recognize when your attention has gone astray and bring it back, right? It sounds a lot like mindfulness in a way, where you need to be able to do two things: stay on task and come back to the task when you've noticed yourself get off track. And so what we're hoping for is that by building in a bit of structure via the biofeedback, people can learn over time to be more metacognitively aware of their own attentive state, and maybe that will have sort of downstream effects on their cognitive performance in really intense and cognitively demanding situations, or even just sitting at their computer all day.
Air traffic controllers,
right.
Think about them, yeah.
Yeah, so I have a colleague at UTA who is an aviation psychologist in the IO area, and we've been having conversations about air traffic control as a test bed for these kinds of ideas, because she was telling me that the big issue with air traffic control is not the people who are at the busy airports, it's the people who are at the slow airports, the ones that are only getting a plane every 45 minutes. And when that's happening, it's much easier to let your attention slide or even literally fall asleep at the wheel, so to speak, and allow something bad to happen. So my collaborators at the Navy, they have pilots, air traffic controllers, and then they also have the mechanics and the ordnance men working there. And so we're measuring cognitive abilities across all these different populations, but what we're sort of specifically interested in is these, how important these abilities, these intensity and consistency things are for those people that are going into those really high demand roles like ATC and piloting aircraft.
So my second question, and then I have a third one, but I'll wait and see who else has questions before I ask my third one. But the second one is, do you feel like this measure could be a measure of response to, for example, when someone gets an ADHD diagnosis and their medication, that, some people respond, sometimes it quits working. Do you feel like this could be a neurophysiologic measure of my response to a drug, of when does it work, when doesn't, is possible?
Yeah, I don't think I've given that specific question much thought, like responsiveness to drugs. We have been trying to find, drugs that we think might selectively affect the norepinephrine system so that we can causally manipulate the system. Lots of drugs, stimulants that are used in these kinds of work seem to be pretty promiscuous and affect both noradrenergic systems, dopaminergic systems, cholinergic systems. So it's really hard to parse apart the relative contributions of each neuromodulator with something like a drug trial. But yeah, that's an interesting question and not one I think I've given much specific thought to. When I was in my postdoc, we were, I wrote an NRSA on applying this to ADHD. So I was reading a lot of Ian Robertson's work, his neurodrenergic theory of ADHD, and some of the other proposals that implicate the neurodrenergic system as opposed to the dopaminergic system, which is often the candidate mechanism that people... So we wanted to do this kind of neuroimaging on ADHD populations versus control. We wanted to bring them in and do these lab-based tasks and get pupillometry measures. Didn't get the NRSA, so that project kind of fell by the wayside, but I got a job instead, so it all worked out. Yeah.
Cool, I have a question. So before, kind of as you were showing the difference between those that have consistency versus being very inconsistent, my first question was, like, were they just more tired, or how does that relate to sleep? And then you said that's something you want to look into. And then I was just curious about, I know you said you're looking into more of that individual variability, but is that, like, does time of day matter, and how consistent is that within someone if you brought them in today versus tomorrow in terms of consistency?
So I'll answer both questions. So this is an obvious thing that comes up in reviews, right? Like, is this just motivation? Is it just fatigue? Like, how do you account for those effects? And so in my my dissertation project, the psychomotor vigilance data that I just presented, we actually asked people to self-report their alertness level and their motivation level 5 times throughout the task. And as you can imagine, motivation crashed, alertness crashed, and individual differences in motivation and alertness correlated with performance and with these evoked pupillary measures and the consistency measures. I didn't show that today because I just felt like it was a little bit too much to try and cover. So recently what I did to follow up on that was I did sort of latent variable analysis of this, and I had people do multiple attention tasks and extracted these features from all 3 tasks. And then I asked them before and after every task, how motivated were you, how alert were you? And like I was mentioning, the correlations there are actually quite weak and do not account for the relationship. So I think this is a pretty stable individual difference. To answer your second question, we just wrapped data on a test-retest study of these things. Luckily, we were able to get back to a project that got paused in March 2020. We wanted to have about 250 participants, and we had about 120 at the time. So we have 250 people's data worth. I just received word yesterday that we got to that number, and so now we can start analyzing the latter half of the data. But in the first half of the data, with the 120 people, so like resting pupil diameter, just measuring my pupil as I'm doing nothing, remarkably consistent week to week. So these sessions were separated from anywhere from 6 to 10 days. We didn't care about time of day. We actually kind of wanted people to come in at different times of day, so that wasn't a confound. The correlation between like day 1 pupil diameter and day 2 was like 0.85, huge, right? So the other thing we were interested in is whether in a task, those intensity and consistency measures were correlated. And now, if I had done the study again, I would have given them the same exact task on both days, but I actually gave people different tasks on different days. But the correlation between consistency and intensity measures within tasks, across tasks, was not moderated by the day. So it wasn't the case that like 2 tasks that were measured on day 1 correlated much stronger than tasks that were, they all correlated like right around 0.35. So again, the case we're gonna make is that this is actually a pretty stable individual difference. Now we did collect data we always collect data on how much sleep did you get last night? Have you consumed any caffeine in the last 4 hours? Did you drink any alcohol in the last 24 hours? A lot of people probably lie on that question. And nicotine, things like that. So we want to see if those things could push or depress your pupil diameter. The problem with these data, we collect them in college students, and almost no one says they're using nicotine. No one says they're using alcohol. No one says they had any caffeine. And so you're comparing sample sizes of 20 to 230, and it's just noisy. So I think certainly that those things probably do play a bit of a role, but a lot of this is pretty stable within an individual. Yeah. So we always ask, so the question was, do we ask about stimulant use like Adderall or something like that? We always ask, are you taking any medications that you believe would affect your memory or speed of response? And so a decent amount of people will say yeah, I'm on Adderall, or I don't think a lot of people, I think people are hesitant to report it if they don't have a prescription for it. And so, maybe some of those responses aren't very truthful. But again, I think 90% of our people say no. So we end up with when we try to compare them, it's hard. Yeah. we'd probably be better off like trying to recruit people who are on a stimulant versus not, and then people who are typically on a stimulant and maybe hold them off their stimulant and see what that, but the ethics of that get a little questionable.
Now it is. My question, you spoke briefly about incentivizing your students with a point system. So that kind of made me think about study design. Do you think that their ability to be able to like auto-correct or like kind of be more consistent throughout would be a long-term effect if that were how you designed the study? And then also on the reverse side of that, do you think that if then there was a cost associated with doing the study, would that potentially affect the results as well?
Yeah, so I don't know about the long-term effects of these manipulations. In fact, I think it might have like a reverse effect if you were to take it away. Like, wait a second, where are my points? We haven't done anything like that. Like we haven't really like given an incentive and then taken it away or added it in halfway through a task because we've got these time on task effects. And all of these are between subject experiments. So we can't really compare across, unfortunately. Now, what was the second part of your question?
If you did the study design with, one you incentivized, the other one if there was a cost to participate in the program, do you think that that would be a motivator for individuals to be more consistent or more intense in their performance?
A cost to participating?
Mm-hmm.
So in my dissertation, one of the experiments was a reward. So we did 3 manipulations. One of them was a goal-setting manipulation. So we told people like a specific number to strive towards on their reaction times, and that helped performance a bit. Didn't eliminate the time on task effect, but mitigated it a bit. Feedback was really powerful. So just telling people at the end of a 20 trial block, here's what your average reaction time was for that block, here's what your average reaction time was overall. People like feedback. They report being more motivated. They mind wander less. They do better on the task. Their task-evoked pupillary responses were larger. That was the most powerful manipulation I actually employed, was just giving people feedback. The third condition I did, which had a kind of interesting pattern of results, but one, I told people, strive towards this goal, and if you hit it, I'll give you a $10 cash bonus. In the other condition, I said, if you strive towards this goal, and if you hit it, you get to leave the experiment a half hour early. And people in that condition said they were way more motivated. They didn't actually do any better. So it was like hard to interpret that effect, but it at least made them. Now that being said, I followed up on that because I was like, why was I not getting a behavioral effect? But I think what happened was I told people to try to keep their average reaction time below 300 milliseconds, which is like really hard. And so I think there was kind of this counteracting influence of like an impossible threshold, and they wanted to get there, but like it just seemed impossible. So what I did instead, and I borrowed this technique from the IO literature, I gave a piece rate incentive. And so I said, for every trial you get your reaction time below 300 milliseconds, you get to shave a trial off at the end. So if you do really well, you'll get out in half the time. Otherwise, the task is going to take an hour. And so that totally, I think it totally got rid of the vigilance decrement, that change in performance. It got rid of, it increased the task-evoked response, it increased performance. So yeah, that's a bit of a punishment. Like you could kind of think about it both ways. Like if I don't do well, I got to be here for another half hour. And like I said, this task is brutal. So like it really does motivate people, especially students. Like all they want to do is get out of the lab. So like it's a really, you don't even have to pay them. Just you can get on your way.
So Matt, let's jump to a couple online questions.
Okay.
So we have some, the hybrid audience has a few things. There's one particular that I want to get to because it's so critical to your talk. Anne Treisman had this idea. She's famous for the binding problem and attention. That like when we went through that color box working memory exercise, in some ways it could be a failure of a binding problem where you don't successfully store location with color. And so what that would suggest is, I guess really the gist of the question is, does the pupillometry effect, which scales with working memory capacity, might it actually still be explainable by attention based on a failure of that binding problem in attention that would then seem to lead to worse working memory?
Yeah, so it's a good question, and it's one that we've been trying to get at behaviorally. We've had a hard time coming up with a good way to measure like encoding, because there's multiple ways to get a trial wrong in that task, right? Like Anne Treisman would argue, you don't integrate the features, and so you don't bind the two important features, color and location, because you only get 250 milliseconds to encode them. And so if you don't encode the items properly, you're not going to remember them. Now you could also, so maybe you're experiencing a lapse of attention at encoding, and in other instances you might be experiencing that lapse of attention during the delay, right? You're sitting there waiting for the test to appear, and all of a sudden you're thinking about the finals. And it seems like there's two potential ways to be, lots of different ways to be a low capacity person, and I think as Randy Engel likes to say, like there's lots of ways to be a low span. Like they call these people, low working memory span peoples. And so, you can be a high span for pretty much one reason. You have to be good at all the aspects of the task. You have to encode the items, remember the items, do well on the test. But you could miss or be wrong on a trial for any given number of reasons. So using pupillometry there is kind of difficult because we don't actually use the pupil to measure encoding like you would in an EEG. Now, I think this is where something like EEG would be really helpful, where you can actually use something like an N2PC that happens at the encoding window that, if that isn't, they don't even look like they've selected the items first of all. So I did a project a couple years ago with Ed Vogel and Kirsten Adam when they were at the University of Oregon with me, looking at these things. So there's EEG signatures of encoding information into working memory and then storing that information. And one of the things that we found was that most attention lapses, working memory lapses, trials where you can't remember the items, are actually storage failures and not necessarily encoding failures. So, as far as individual differences go, I should know the answer to this, but I can't quite remember what we found as far as individual differences. But I will say this study was not quite designed to look at individual differences. It was more designed to look at sort of success versus failure and what causes that on a trial-by-trial basis, yeah.
All right, we are about at time. Let's go ahead and thank Matt Robison. You are consistently intense throughout this whole talk.
All right, thank you.
There's so much to learn about and think about here. Before we leave, we will I want to remind the online audience to please take note of the BrainHealth Project. This is our landmark study. If you are interested in learning more about this, please visit the website for the center, or you can scan the QR code, which will be on screen. This is it as far as the spring season of Frontiers. Thanks so much for joining us, and we will be back with another great lineup starting in August, and we'll see you then.